This project is on Minamata disease. Minamata is a small inlet on the southwestern coast of Kyushu Island in Japan. In 1953, there was an outbreak of a mysterious illness in Minamata children and cats. It was locally called the dancing cat disease because of the strange, erratic behavior ill cats exhibited. Soon enough, though, adults began showing the same symptoms of what was assumed to be a neurological disease, and the epidemic was officially recognized in 1956. Symptoms included impairments of vision and hearing, changes in sense of smell and taste, dysarthria or inability to speak, ataxia or inability to walk normally, several and severe psychiatric disorders, paresthesia or numbing, tingling, and hypersensitive sensations of the extremities and lips, and a crippling convulsion disorder, similar in appearance to epileptic events. Because the outbreak initially appeared contagious, it was assumed that its cause was bacterial or viral, and a quarantine was placed. However, as the disease continued to spread and affect both humans and animals, people looked to the nearby Chiso chemical factory. The Chiso factory was located just upstream from Minamata, and during the Second World War, began dumping massive amounts of waste into the Minamata River, contaminating the fish, which were a primary source of food for the citizens. Once it became obvious enough that the source of the outbreak was the Chiso chemical factory waste, a new problem presented. The Chiso factory was discharging many metals, so which one was responsible for Minamata disease? The question remained unanswered for years until a doctor recognized that the symptoms of Minamata disease closely resembled those of mercury poisoning. Mercury poisoning, like Minamata disease, is a neurological illness. This is a comparison between a normal brain on the left and a brain damaged by chronic mercury poisoning on the right. But how does mercury damage the brain? For starters, every cell has a cytoskeleton made of filaments and microtubules to give it structure and stability. Microtubules are made of subunits called tubulin, but mercury binds to tubulin in the place where GTP is meant to bind, inhibiting further synthesis of the microtubule. As a result, the cell is unable to continue synthesizing its own skeleton, and it collapses on itself, resulting in cell death. This process occurs in nerve cells, which, unlike most other cell types, are unable to be replaced once they're killed. Consequently, the damage that occurs as a result of mercury poisoning is permanent. But how can we be sure that Minamata disease is caused by mercury poisoning? There are several reasons. Researchers measured the amount of mercury in afflicted patients' hair, blood, and in contaminated fish, and in each case, the results showed astoundingly high concentrations of mercury. Turns out the Chiso factory was using mercury as a catalyst. The first reaction shows the synthesis of a catalyst called Denagis reagent, which in the second reaction is used to synthesize acetaldehyde, an important compound for the later synthesis of plastic materials. This process never presented a problem until Chiso switched the co-catalyst from manganese dioxide to ferric sulfide. After introducing the new co-catalyst, the production of methylmercury as a byproduct increased. The methylmercury was then released into the water, where it contaminated the fish and the citizens of Minamata. Once the cause of Minamata disease was pinpointed to the production and release of methylmercury, the Chiso factory moved its waste discharge to Hyakin Harbor, mainly to avoid investigators. However, this only resulted in further spread of the disease. Then Chiso attempted to, but failed, to clean the water it was contaminating. So to this day, there are fishing bans on the coast of Minamata Bay. So what are some possible remediation strategies for this devastating event? In class, we discussed the use of chelating agents, particularly ethylene diamine tetraacetic acid, or EDTA. Chelating agents are compounds that bind strongly with metals, facilitating removal of those metals from solutions. A chelating agent currently used for treatment of mercury poisoning is British anti-leucite, or BAL. However, two other chelating agents have been proposed for treatment of mercury poisoning, dimercaptosuccinic acid and 2,3-dimercaptopropane 1-sulfonate, or DMSA and DMPS respectively. The problem with our current agent is that BAL has a low LD50, meaning that it takes a relatively small amount of BAL before it itself becomes toxic to the patient. On the other hand, DMPS, DMSA, and EDTA have a higher LD50. However, as discussed in class, EDTA needs to be fully deprotonated before it can become useful. As a consequence, it's only useful in solutions with a high pH. As you can see, the fraction of dissociation for the fully deprotonated useful form, abbreviated as Y4-, is only reasonably high in a pH of at least 9. The pH of the Minamata River and of human blood is not high enough for EDTA to be effective. On the other hand, trials have been conducted on poisoned rats and mice, and the results show that DMSA and DMPS are still effective in binding to mercury, even in relatively neutral environments, with DMSA being the most effective. Thus, DMPS and DMSA may be a more efficient and safe means of treating Minamata disease. So yeah, that's Minamata disease for you. And here are our resources.